Good evening. Good evening, everyone. Yes, Symmetry, Fabio, Ash, Harsh, good evening, beta. Good evening. So today we are going to discuss a very important pathway, okay, that is HMP pathway or commonly known as the hexose monophosphate pathway. So before starting with the session, Abrar, good evening, beta. Good evening. So before starting with the session, beta, just give me a thumbs up in the chat box if the audio and visuals are perfectly fine. If the audio and visuals are perfectly fine, just give me a thumbs up in the chat box so that I can start with the session. Yes, Harsh, very good, beta. Yes, Chenam, very good, beta. Abrar, very good, very good. So kindly keep commenting in the chat box as well simultaneously so that it uh, okay so that yeah, yeah, good evening, Ashish. Good evening. I hope I'm not teaching the walls over here, so I want everyone to reply in the chat box. Yeah. So let us start with the HMP pathway. Let us start with the HMP pathway. Now, firstly, what is the full form of HMP pathway? What is the full form of HMP pathway? Achal Chaudhary, good evening, beta. Good evening. So yes, let us start with HMP pathway. HMP pathway. Now you already know beta. HMP stands for okay. HMP stands for hexose monophosphate pathway. Hexose monophosphate pathway. Now we have another name for this. It is hexose monophosphate pathway. Yes, Ashish, very good. Chandru, very good, beta. Good evening, Chandru. Very good, beta. Hexose monophosphate pathway. It is also known as pentose phosphate pathway. It is also known as pentose phosphate pathway. It is also known as pentose phosphate pathway. Okay, it is also known as pentose phosphate pathway. Right? Apart from that, it is also known as Dicken because they were proposed by the scientist. It is also known as Dicken-Horecker pathway. Okay, it is also known as Dicken-Horecker pathway. So we have three names, one based on the name of the person who has discovered it. Yes, Usman, good evening, beta, good evening. And the other two, yeah, it is the hexose monophosphate pathway or pentose, yeah, phosphate pathway. Now, beta, before starting with this, firstly, you have to remember where this HMP pathway takes place. So, yes, so you have to remember what is the site. Okay, what is the site? Okay, so yes, site I will come, but yeah, this HMP pathway is taking place. Okay, is taking place all over. Okay, I will come to the different phases. Yes, I will come to the different phases. Yes, I will come to the difference also, Chandru. I know you are going to ask me this question that what's the difference between HMP pathway and glycolysis. I will be coming to that question also. Yeah, at the end. So no need to worry. Yeah. HMP pathway it is taking place in cytosol or yes, it is the cytoplasm. Okay, cytosol or we commonly call it as the cytoplasm of the cell. Okay, only in the cytoplasm. Okay, only in the cytoplasm of the cell. Now which organs are involved? Okay, which organs are involved I will be teaching okay in the respective phases. So you already know that we have two steps, okay, or two phases of HMP pathway, oxidative and non-oxidative, and non-oxidative. So before coming to that, what is the significance? Okay, now what is the significance of this pathway? Can anyone tell me in the chat box what is the okay, what is the product that we are getting from this pathway? What is the product that we are getting from this pathway? Anyone? You already know from this pathway, from hexose monophosphate pathway beta, we are getting two products. Okay, we are getting mainly two products beta. The very first one, okay, the very first one, it is NADPH. NADPH, nicotinamide adenine, okay, dinucleotide phosphate hydrogen. Okay, we are getting this product from this HMP pathway. Okay, so you already know it is nicotinamide adenine dinucleotide phosphate hydrogen. Phosphate hydrogen. Yes, yes, Usman, very good, beta. Yes, Usman, very good. And Ashish, you also are right. You also, like, you have written correct. It is fructose 6 phosphate also. But yeah, the major products I'm just talking about over here: nicotinamide, adenine, dinucleotide, phosphate. Yeah, phosphate hydrogen. Okay, it is phosphate hydrogen. Yes. Now, beta, remember the major source. Okay, the major source for NADPH. This question has been asked. The major source, you are getting NADPH in the body, okay, from glycolysis also, other pathways also, but the major source of NADPH, it is HMP pathway. It is HMP pathway. Yes, it is HMP pathway. Now, now F Chandru has written, yes, cholesterol synthesis. Cholesterol synthesis. Now, beta, remember, yes, this NADPH, okay, this NADPH, yeah. Now, this NADPH has various uses, right? I will come to the significance of NADPH, but yes, remember, beta, it is used for the biosynthesis of fatty acids, okay, biosynthesis of fatty acids, steroids, cholesterol, as Chandru already told. Okay, cholesterol. Okay, now these are the organs where no the oxidative phase can operate. For example, liver, gonads, ovaries. Okay, adipose tissue. So basically, they are major source of NADPH. Okay, they help in removal of free radical. I will be coming to all the points. No need to remember. They help in removal of free radicals. Okay, free reactive species. Yes, the free radicals. Okay, removal of free radicals. Removal of free radicals. Okay, apart from that, they help in killing of bacteria also. Okay, they help in killing of bacteria also. You already know it helps in phagocytosis. Okay, yes, it helps in phagocytosis, right? It helps in phagocytosis. Yes, steroid hormone synthesis. Yes. So major source is NADPH. One of the like many significance I have just written on the screen. Apart from that, Peter, apart from that, you have to be clear that there is one more product that you will get, that is ribose. Okay, that is ribose 5 phosphate. Ribose 5 phosphate, right? Ribose 5 phosphate. Now, this ribose 5 phosphate, okay, this ribose 5 phosphate, it is actually necessary, okay, it is actually necessary for formation of, okay, for formation of RNA and DNA, for formation of RNA and DNA, right? So yes, it is, yeah, it is a precursor for purines and pyrimidines. Yes, Kriti, very good, beta. And remember, it is necessary for the formation of RNA and DNA. So basically, two sources. Okay, what is the significance of HMP pathway? Beta, remember, it is the major source. Okay, it is the major source of NADPH. Okay, major source of NADPH, right? And NADPH, you already know, it is used in variety of reactions and purposes. Apart from that, it is a contributor of pentosis. Okay, it is a contributor of pentosis, beta, and you already know these are actually these pentoses. These are responsible for the formation of DNA and RNA, right? We have like yes, or ribose 5 phosphate. You can just write it is over here. It is contributor of pentosis. Okay, contributor of pentosis. And these pentoses are basically necessary for the formation of RNA or DNA. Right? I hope this is clear to each one of you. Better just give me a thumbs up in the chat box. Just give me a thumbs up in the chat box if this is clear to each one of you. Yeah, if this is clear to each one of you, just give me a thumbs up in the chat box. Yes. Sure. Very good. Harsh, Ashish, Ash, Usman, Jainam, very good. Very good, beta. Very good. Very good. Very good. Kriti, very good, beta. Very good. Chalo, let us now start. Yeah, let us now start with the steps or the phases of HMP pathway. We have discussed the site, okay. We have discussed the significance. We have discussed, okay, the importance of NEET-PG and these pentoses, right? Now let us now start with the steps. Okay, let us now start with the steps or phases of HMP pathway. We have steps or Varun, very good, beta. Varun, very good. Steps or phases of HMP pathway. Phases of HMP pathway. So remember, beta, we have two phases of HMP pathway. The two phases of HMP pathway. We have oxidative phase. We have oxidative phase, and we have non-oxidative phase. Non-oxidative phase. We have oxidative and non-oxidative phase. Yes, I will be discussing the reactions, Chandru. Don't worry. Absolutely don't worry. So yes, now the very first one beta that you have to remember over here. Yes, it is glucose 6-phosphate. Glucose 6-phosphate. You already know from glucose what is formed. From glucose what is formed? Glucose 6-phosphate. From glucose what is formed? Glucose 6-phosphate. Now I want everyone to answer the enzyme which is responsible for the conversion of glucose to glucose 6-phosphate. चलो फटाफट से बता दो इसमें कौन सा एंजाइम यूज होगा जल्दी से चंद्रू आई थिंक योर आंसर विल बी हियर राइट नाउ हेक्सोकाइनेस यस yes Ashish Lohar very good beta it is the hexokinase which is responsible over here yes everyone is right Usman Kriti Janam very good beta it is the kinase enzyme. So remember we have hexokinase, and there is utilization of an ATP molecule. Okay, ATP will be utilized in the presence of magnesium 2 plus ions. Magnesium 2 plus ions. Yes, everyone is right. Harsh, very good. Ash, very good, beta. Very good. Now you already know this glucose 6 phosphate, okay, will enter the oxidative phase. So I will be first discussing about, okay, the oxidative phase. Okay, oxidative phase. Oxidative phase. Since everything is happening in the cytoplasm, now this glucose 6 phosphate beta, this glucose 6 phosphate, yes, yes, beta, glucokinase and hexokinase. Obviously, beta, have I like told you the difference between the hexokinase and glucokinase? Hexokinase, beta, it is like a homemaker. Homemaker यानी कि housewife की तरह है. She will work like as the housewife works continuously without any rest. और किसी भी सूरत में this is just like with the hexokinase. Okay. Glucokinase कभी कभी no active होता है in special circumstances. Okay. That is why yeah Chandru is writing about the liver and pancreatic beta cells, right? So yeah. तो वो मेहमान की तरह है ग्लूकोकाइनेस और एच हेक्सोकाइनेस इज जस्ट लाइक द एच से याद रखो हाउसवाइफ जो पूरा दिन काम करती रहती है ठीक है तो बेसिक तो ये अभी समझ लो बाकी अभी और भी डिफरेंसेस लिखवा दूंगा मैं ओके नाउ बेटा रिमेंबर दिस ग्लूकोज 6 phosphate will convert okay will convert into 6-phosphogluconate. 6-phosphogluconate. Okay, 6-phosphogluconate. Okay, now remember beta, glucose 6 phosphate is also a 6 carbon compound. It is also a 6 carbon compound. Okay, now what is happening over here? Yeah, oxidation is taking place. Oxidation is taking place. Yes, with the help of enzyme, glucose 6 phosphate dehydrogenase. Dehydrogenase, right? Glucose 6 phosphate dehydrogenase. Remember beta, yes Sanjay, very good beta. It is the rate limiting enzyme. Okay, it is the rate limiting enzyme. Second important thing that you have to remember beta over here, this is an irreversible reaction. Okay, this is an irreversible reaction. This is irreversible reaction. Okay, this is an irreversible reaction. Now, from glucose 6 phosphate, what is formed beta? 6 phosphogluconate. Okay, and since the enzyme glucose 6 phosphate dehydrogenase, yes, what it will form? NADPH. NADPH. NADPH plus H plus. Okay, and it will be formed from NADP plus. NADP plus will form NADPH plus H plus. It will be oxidized to form NADPH plus H plus. So you can see the major source. Okay, the major source over here, you can see it is NADPH. Okay, we are getting NADPH. I will be coming to G6PD deficiency beta. अभी सारे क्लिनिकल कोरिलेशंस भी कराऊंगा. अगर हेडिंग पढ़ी होगी आज के जो थंबनेल पर, वो सब लिखा हुआ है, सब पढ़ेंगे हम लोग. 6-phosphogluconate, 6-phosphogluconate will convert into, okay, it will convert into ribulose, ribulose 5-phosphate, ribulose 5-phosphate. Now my question to you is, ribulose, it is aldehyde derivative or keto derivative? चलो इसका आंसर तो ये मैंने पहले ही लेक्चर में बताया था. I have already told you in the very first lecture students. राइबोस और राइब्यूलोस, फटाफट से बता दो, ये एल्डिहाइड डेरिवेटिव है, कीटो डेरिवेटिव है, जल्दी से. ये तो पता है, बट आई वांट टू नो यू गाइस नो दिस और नॉट. यस, आशीष फेस कीटो. व्हाट अबाउट रेस्ट गाइस? व्हाट अबाउट रेस्ट? Ribulose 5-phosphate, obviously it will be a 5 carbon compound. So from the 6 carbon, 5 carbon is there, so there will be loss of a CO2 molecule, loss of a carbon in the form of CO2. Yes, Jainam, very good. Usman, very good बेटा. Ash, Kriti, Harsh, very good बेटा. चलो, थोड़ा बहुत याद है तो बिल्कुल ठीक है. और again this is a, yeah, oxidation taking place over here. So remember beta, the enzyme will again remain the same. 6-phosphogluconate, 6-phosphogluconate dehydrogenase, dehydrogenase, and you already know from NADP plus, from NADP plus what will be formed? NADPH plus H plus, right? It will be oxidized to form NADPH plus H plus. So yes, that is again you are getting NADPH in both the reactions. And remember, like this is done with the oxidative phase beta. Oxidative phase हो गया यहाँ पे, oxidative phase हो गया, and you can see we are getting NADPH. We are getting NADPH. Now, what is the significance of NADPH? Or in other ways, if I want to write, if I have to like link biochemistry with the medicine beta, remember you just don't have to remember the reactions, about the significance also, as I always link the reactions with the clinical part. So yes, what are the functions of NADPH? Functions or significance? Functions or significance of NADPH? Functions or significance of NADPH, right? NADPH. Yes. Now, remember beta, yes, the very first one that you have to remember, It helps in the reductive biosynthesis. Reductive biosynthesis of fatty acids and steroids. Fatty acids and steroids. Okay. Cholesterol. Cholesterol. Okay. And cholesterol. So it helps in the reductive biosynthesis of fatty acids, steroids, cholesterol. Okay. And organs. Again, you have to remember the organs where oxidative phase. You already know we have two phases, oxidative and non-oxidative phase. And the organs where oxidative phase operates. Remember, the very first one, it is the liver. The very first one, it is the liver. The second one, it is the adipose tissue, the fat tissue, right? The third one, we have the gonads. Yes, gonads. Yes, and like ovaries also. Ovaries also. Yes, we have lactating mammary glands, lactating mammary glands, and apart from that, beta, we have the thyroid glands. Thyroid glands. Okay, so we have liver, adipose tissue, gonads, lactating mammary glands, and thyroid glands. These are the places where the oxidative phase, okay, is functional, right? Or where it operates. These are the major organs, beta. I hope this is clear to each and everyone. I hope this is clear to each and everyone. Just give me a thumbs up so that I can discuss with the other significance of NADPH. So this was the first one. Yes, Ash, very good, beta. Harsh, very good. Ashish, very good, beta. Very good. Now the second one, beta. The second one. Yes. Usman, Kriti, very good. Very good. Now, the second one, you have to remember that NADPH, okay, it is used for free radical scavenging. Free radical scavenging. Now, beta, free radical scavenging. We have the reactive species, beta. Before they can affect the cell, okay, before they can affect the cell and cause cancers or can cause any other damage to the cell, these free species, okay, or free reactive species are removed by this NADPH from the cell. अगर वो सेल के अंदर रहेंगे बेटा अगेन दे विल कॉज समथिंग लाइक कैंसर्स दे कैन कॉज ट्यूमर्स राइट दे कैन कॉज हार्म टू द सेल एंड before they cause any harm to the cell these reactive species are removed from the cell with the help of NADPH or in turn these reactive species are not formed also both ways are right okay both ways are right ये एक तो पहली बात NADPH it doesn't allow these reactive species to form or in case if they are formed also they are being removed from the cells before they can do some action and harm the cell right so yes you have to remember beta you already know can anyone tell me the name of this compound that I have written over here H2O2 को क्या बोलते हैं yes it will be oxidized over here yes tell me बेटा जल्दी से बताओ चलो जल्दी से बता दो फटाफट से hydrogen peroxide. What about rest? We have GSH, it is the glutathione. Yes. हाँ ठीक है आशीष ठीक है संजय ठीक है जीएस चलो जीएस मैंने बता दिया जीएस एसएच बताओ क्या होता है जीएस एसएच बताओ क्या होता है हाँ चलो ठीक है हाँ ठीक है बेटा GSH it is glutathione right it is glutathione or GSSG it is disulfide glutathione right now this is like this And remember beta, yeah, this, yes, NADPH is reduced to form this NADP. It is reduced to form NADP in the help with the help of an enzyme over here. Yeah, that I will write with another pen. Yeah, this is okay. FAD, in the presence of FAD, and the enzyme, it is glutathione reductase. Okay, over here we are using glutathione reductase. Okay, and over here it is oxidized, so this is the oxidized form. Okay, this is the oxidized form. Okay, and over here, yeah, if it is getting back, yeah, it is glutathione peroxidase. Glutathione peroxidase, right? Glutathione peroxidase. So you can see that how NADPH, it is basically utilized to know oxidize these free radicals H2O2. Okay, the glutathione. Okay, glutathione is basically formed. Okay, it is oxidized to form glutathione disulfide glutathione. Okay, disulfide glutathione, right? Or yeah, I can just write it as glutathione disulfide. So obviously they are non-reactive. So this is how this NADPH, no, it is using or it is needed for free radical scavenging. Okay, उनको पकड़ के खत्म कर देना. Okay, so it's just change the nature of these so that they are non-harmful to the cell. I hope this is clear to everyone. I hope this is clear to everyone. Yeah. Now, why free radical scavenging is important? But why free radical scavenging is important, beta? Remember, yes, yes, Harsh, Ash, Ashish, Usman, very good. Why free radical scavenging is important, beta? You have to remember that it is important in RBC. Okay, it is important in RBC to maintain membrane integrity of RBC, to maintain the membrane integrity of RBC, to maintain the membrane integrity of RBC. Apart from that, beta, apart from that, yes, the second most purpose over here, yes, free radical scavenging. It is important in RBC. It is also important, okay, in lens to keep the transparency of the lens, to keep the transparency of the lens, right? So yes, free radical scavenging, it is important in erythrocytes, okay, in RBC to maintain the membrane integrity of the RBC, to in the lens to keep the transparency of the lens, and even if we talk about the hemoglobin beta, hemoglobin, okay, Fe2 plus state, if it is converted to methemoglobin, so it will be like Fe plus 3. And yes, it does not allow the normal hemoglobin to convert into methemoglobin. Now methemoglobin beta, you already know it is a reduced state. Okay, it is a reduced state. Okay, so yeah, so it helps. Oh, sorry. Yeah. Okay, it helps the hemoglobin to be in this state, right? So yeah, that is something that you have to remember about the free radical scavenging. It is like important in erythrocytes to maintain the membrane integrity, to keep the hemoglobin, okay, in Fe plus 2 state. It does not allow the hemoglobin to move to methemoglobin. Apart from that, it keeps the transparency of the lens. Okay, it helps to keep the transparency of the lens. That is all about the second part. That is all about the oxidative phase beta. That is all about the oxidative phase. Now my question to everyone over here. हाँ, किलिंग ऑफ बैक्टीरिया भी लिख सकते हैं बेटा, कोई दिक्कत नहीं है. या किलिंग ऑफ बैक्टीरिया भी है. you already know the neutrophils. Okay, अगर तुम्हें पता ही है neutrophils. Okay, मैंने ये वैसे लिखवा दी है तुम लोगों को. I suppose I have already discussed this. या killing of bacteria आप यहाँ पे भी mention कर सकते हैं बेटा. Neutrophils जो होते हैं ना, this NADPH in the neutrophils, it helps in the killing of bacteria. Okay, one more point you can add if you want, right? हाँ, चलो. I hope. Okay, if I ask you a question, what is the end product of the oxidative phase of HMP pathway? What is the end product of oxidative phase of HMP pathway? चलो फटाफट से आंसर दो. ये तो सबसे इजी क्वेश्चन है. एंड प्रोडक्ट क्या बन रहा है बेटा? स्टार्टिंग कहां से हुई थी? ग्लूकोज 6 फॉस्फेट से, राइट? और एंडिंग कहां पे हो रही है? राइबुलोज 5 फॉस्फेट पे. सो द नॉन ऑक्सीडेटिव फेज, yes, very good Harsh, very good Ashish, very good. So the non-oxidative phase will start from ribulose 5 phosphate, right? So the non-oxidative phase, chalo, let us start with the non-oxidative phase. Yes, let us start with the non-oxidative phase. Non-oxidative phase. Now, yes, ribulose 5 phosphate, ठीक है. चिराग, कृति, उस्मान, very good बेटा. Ribulose 5 phosphate, बिल्कुल ठीक है. So now we will start with the non-oxidative phase. Now before, you should okay, we start with the reactions beta. Remember, this is a reversible phase. Okay, it is a reversible, okay, a phase which is reversible. Okay, and over here you will get a product that is called as the pentosis. Okay, pentosis. Yeah, yes, we have these pentosis which we will get, and you already know these pentoses are required for DNA and RNA synthesis. And this non-oxidative phase beta, non-oxidative phase, basically it occurs in all organs, and specifically if we talk about specifically in, remember beta, it is the skin. Okay, it is the skin, intestinal mucosa, and bone marrow, So it is occurring in all organs. Specifically, it is occurring in skin, intestinal mucosa, and bone marrow. Okay. So yeah, these are the skin. You already know it is the largest organ of the body. So we have rapid cell turnovers over here, right? Intestinal mucosa, bone marrow. Now coming on to the steps. Or the reactions. Steps of non-oxidative phase. Steps of the non-oxidative phase. Now, this reaction will be started from ribulose 5-phosphate. Okay, this reaction will be started from ribulose 5-phosphate. You already know we have ribulose 5-phosphate. So I'm taking 3 molecules, beta. I'm taking 3 molecules over here. Ribulose 5-phosphate. And we have ribulose 5-phosphate. Ribulose 5-phosphate. So we have just taken 3 molecules. Okay, ribulose 5-phosphate, ribulose 5-phosphate, ribulose 5-phosphate, because it was the end product in the oxidative phase. We have already seen this. Now this ribulose 5-phosphate, okay, you already know it is a 5 carbon compound. It is also 5 carbon, and like I have taken 3 molecules, the same molecules are there. You already know these are 5 carbon compounds. Now this ribulose 5-phosphate, will change, okay, will change into ribose 5 phosphate. Now, this is also 5 carbon beta. This is also 5 carbon. You guys are going to tell me which enzyme will be used over here. You guys are going to tell me which enzyme will be used over here. I want answers from everyone in the chat box. Yes, I want answers from everyone. Yes. हाँ चलो जैनाम वेरी गुड हर्ष वेरी गुड इट इज द आइसोमरेज इट इज द आइसोमरेज बेटा ऑब्वियस सी बात है ना 5 कार्बन है ये भी 5 कार्बन है ओके यू ऑलरेडी नो राइब्यूलोज और राइबोज में से एल्डिहाइड डेरिवेटिव कौन सा है विच वन इज एल्डिहाइड डेरिवेटिव गाइस आई वांट द आंसर्स इन द चैट बॉक्स यस उस्मान वेरी गुड यस आशीष ऐश वेरी गुड बेटा विच वन इज एल्डिहाइड डेरिवेटिव बेटा ये मैंने सब पढ़ाया हुआ है उसी चीज को देखो मैंने इसलिए बेसिक्स कराए थे ना ताकि अभी दिक्कत ना हो हाँ राइबोस वाला है बेटा वेरी गुड राइबोस एल्डिहाइड डेरिवेटिव है और राइबुलोस कीटो डेरिवेटिव है राइट और ये सॉरी ये की चलो मैं इधर लिख देता हूँ guys will get confused otherwise right so you already know ribulose जो है yes it is keto derivative right और राइबोस वाला जो है it is aldehyde derivative so you already know the enzyme over here will be isomerase only and most like if I have to name it specifically it is ketoisomerase okay it is ketoisomerase from the keto which group is formed aldehyde group so it is ketoisomerase right everyone is right now beta the other two ribulose 5 phosphate will convert beta it will convert into okay xylulose 5 phosphate now beta ribulose and xylulose are epimers okay they have the different configuration of OH I will come to the epimers also but as of now remember beta they have the different configuration of OH in their structure एक में OH इस साइड पर है एक में OH उस साइड पर है so मतलब they have different configuration so remember beta both over here okay will form yes xylulose 5 phosphate xylulose 5 phosphate so we have different configuration in the OH groups that's why the enzyme over here which will be used it is epimerase okay it is epimerase and it is also epimerase right now remember beta what will happen now this xylulose 5 phosphate okay it is also containing how many carbons 5 carbons right it is also containing 5 carbons and it is also containing ribose 5 phosphate it is also containing 5 carbons now this ribose 5 phosphate okay and xylulose 5 phosphate okay what they will form now beta total we have how many carbons 10 5 plus 5 कितना हो जाता है 10 carbons हो गए now they will split into 3 carbons and 7 carbons अब 3 carbons का जो structure है you already know it is called as glyceraldehyde क्योंकि ये aldehyde derivative है तो क्या बनेगा glyceraldehyde you already know glyceraldehyde is a 3 carbon compound 5 और 5 मिला के कितने हो गए 10 तो 10 में से 3 का क्या बन गया ग्लिसरल्डिहाइड और एल्डिहाइड क्यों बनेगा क्योंकि राइबोस 5 phosphate is an aldehyde derivative right and we have now 7 carbon left and that 7 carbon compound it is called as pseudo heptulose okay pseudo heptulose okay so we have glyceraldehyde 3 phosphate yes and pseudo heptulose 7 phosphate देखो phosphate group तो लगा ही हुआ है ना phosphate group तो लगा ही हुआ है so we have glyceraldehyde 3 phosphate and pseudo heptulose 7 phosphate and remember beta it is a 7 carbon compound So usually, we have ribulose 5-phosphate. Now these ribulose 5-phosphate will change. Okay, the one will change into aldehyde derivative, the other will change, just the configuration of OH will be changed, and they will get converted to xylulose. Xylulose and ribose. Okay, xylulose and ribose, they both have 5 carbons. In total, we have 10 carbons. Out of these 10 carbons, a 3 carbon compound will be formed. That is carbohydrate, glyceraldehyde, the parent carbohydrate, and we 7 left ho gaya, so it will be pseudo heptulose or 7-phosphate. Okay, that is a 7 carbon compound. इतना क्लियर है सब लोगों को? जल्दी से एक थम्स अप दो, जल्दी से एक थम्स अप दे दो। बेसिक मैथमेटिक्स पढ़ाई है अभी तो मैंने, चलो फटाफट से जल्दी से एक थम्स अप दो। एक बेसिक मैथमेटिक्स पढ़ाई है। हाँ, हर्ष वेरी गुड, आश वेरी गुड, जैनम वेरी गुड, Kriti very good बेटा। Now, now, again we have 10 carbons. Again we have 10 carbons. Now, again we have 10 carbons. Chirag, Usman, Sanjay, very good. Now, you already know what they will form now. What they will form now. Now, this glyceraldehyde 3-phosphate will get converted, and the pseudo heptulose, they will form a compound that is called as fructose 6-phosphate। फ्रक्टोज 6-phosphate, ऑब्वियसली बात है 3 और 7 कितने हो गए 10, आउट ऑफ 10 6 carbon compound is fructose, 6 carbon compound is fructose, and how many carbons are left now? 4 carbons beta, remember that will be erythrose, that will be erythrose 4-phosphate। ये मुझे बताने की भी जरूरत नहीं है, erythrose we have already discussed this in the first class, right? Erythrose, okay, erythrose, so 2 compounds will be formed, yes, erythrose 4-phosphate and fructose 6-phosphate, fructose 6-phosphate। Now what will happen beta? This erythrose 6-phosphate and this xylulose 6-phosphate, now remember this erythrose 6-phosphate and yes, this xylulose, sorry, xylulose 5-phosphate, again they will form 2 compounds। ये चार this is like you already know, this is 4 carbon, and xylulose 5-phosphate, it is 5 carbon, so together we have 9 carbons now, together we have 9 carbons now, so they will split into, yes, they will split into glyceraldehyde, it will split into glyceraldehyde, and you already know glyceraldehyde 3-phosphate, it is again a 3 carbon compound, and it will form fructose, it will form fructose 6-phosphate, fructose 6-phosphate, it is a 6 carbon compound. So if you look beta, if you look over here, we have the major products, okay, the major products that you can see over here, the end products, it is fructose 6-phosphate, glyceraldehyde 3-phosphate, and fructose 6-phosphate, fructose 6-phosphate, fructose 6-phosphate। तो फटाफट से मुझे बताओ क्या ये समझ में आ गया सब लोगों को? जस्ट गिव मी अ थम्स अप बेटा इफ दिस इज क्लियर टू ईच वन ऑफ यू, just give me a thumbs up if it is clear to each one of you। Yes, Harsh, Ash, Sanjay, Jainam, very good beta। Yes, so that was about the non-oxidative phase, okay, non-oxidative phase beta। Remember over here one more thing, remember over here one more thing, that no ATP is generated, no ATP is generated, okay, no ATP is generated, all reactions are reversible, all reactions are reversible, and yes, one more thing, that is, yeah, now I want to teach one more thing beta over here. So you already know we have, yeah, fructose 6-phosphate, fructose 6-phosphate, just I'm copying this last reaction, just I'm copying this last 3 end products that we have like we got in this particular reaction. So we have fructose 6-phosphate, we have glyceraldehyde, okay, let me correct it, yeah, we have glyceraldehyde 3-phosphate, yes, we have glyceraldehyde 3-phosphate, and we have fructose 6-phosphate. Fructose 6-phosphate. Now, beta, this fructose 6-phosphate will change into glucose 6-phosphate. It can change into glucose 6-phosphate, right? By the help of which enzyme you guys are going to tell me? By the help of which enzyme you guys are going to tell me right now? Glyceraldehyde 3-phosphate. It is a 3-carbon compound, beta. It will also change into glucose 6-phosphate. Okay, it will also change into glucose 6-phosphate. Yes, yeah, like half of the glucose 6-phosphate will be formed. Now, fructose 6-phosphate to glucose 6-phosphate. Isomerase. Jainam is saying isomerase. Obviously, beta, ye yoga. Yes, very good. Even I don't need to tell you, my students are very much, yeah, yeah. Very good, guys. Yes. Now over here, beta, remember if we have like two molecules of this glyceraldehyde 3-phosphate, what will happen? If half glucose 6-phosphate is formed, there will be liberation of 3 CO2 molecules. Okay, so yes, 3 CO2 molecules are like given in oxidative phase. Okay, and this fructose 6-phosphate will again change into glucose 6-phosphate. Yes, glucose 6-phosphate. So yes, CO2 is liberated by this pathway. No ATPs are generated. All the reactions are reversible. All the reactions are reversible. So I hope till now everything is clear, beta. I hope till now everything is clear. Just give me a thumbs up in the chat box if it is clear to each and everyone. Yes. Now coming on to the clinical correlations. Now coming on to the clinical correlations, Harsh, Jainam, Ash, and this is something very important that you have to remember. The clinical significance or the clinical correlations. The clinical correlations. Now remember beta, the very first thing, remember over here. Yes, most common, MC means most common enzyme deficiency. Most common enzyme deficiency in humans. Most common enzyme deficiency in humans, it is glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase deficiency. Now, if this enzyme is deficient, what it will lead to? There will be decrease in amount of NADPH. There will be decrease in amount of NADPH. You all agree with the fact because it is NADPH, the major source. It is the pentose phosphate pathway. It is the pentose phosphate pathway. Now you already know if the NADPH concentration will decrease. Now what it can lead to, beta? What it can lead to? There will be decrease. There will be decrease in free radical scavenging. There will be decrease in the free radical scavenging, right? Apart from that, beta, what will happen? Yeah, since it is taking place in the RBC will undergo hemolysis, okay? Hemolysis, and it is most likely that the patient will suffer from hemolytic anemia. Okay, you will be discussing these in pathology. Hemolytic anemia, right? Okay, it can also lead to jaundice. Okay, it can also lead to jaundice. It can also lead to jaundice. It can also lead to jaundice. Okay, and you already know we have discussed one more point. Hemoglobin will be now converted to meth hemoglobin. Okay, meth hemoglobin. Now, there are certain drugs. Okay, if a patient is suffering from this particular enzyme deficiency, it is always mandatory beta कि उन patients को हम लोग ये drugs नहीं देंगे. We will not give these drugs to these patients, and you already know these drugs are sulfur drugs. Okay, these drugs are sulfur drugs. We have primaquine. we have prima queen, okay, we have fava beans, fava beans. Now, these things are avoided in the patients who are suffering from glucose 6 phosphate dehydrogenase deficiency. So, what they do basically, they will aggregate, okay, they will aggregate, okay, or aggravate your बढ़ा देते हैं, aggravate, yes, G6PD deficiency, ये और बढ़ा देंगे, complications और ज्यादा बढ़ जाएंगे, ठीक है. Now remember beta, fava beans will lead to something which is called as favism, and you guys are going to tell me in the next class about this, right? Yes. Apart from that, one pathology question that has been asked from this particular topic beta, that Haynes body, okay, Haynes bodies, okay, when you will see the RBC, okay, what you will see Haynes bodies, I don't have the picture with me, so today after the class you will see in the Google what exactly these like how exactly these Haynes body looks like. So Haynes bodies in, yeah, yeah, in RBC you will see, and remember it is a feature of, if you are able to see this Haynes body in the RBC, remember this is a feature of G6PD deficiency. It is a classical feature of G6PD deficiency, glucose 6 phosphate dehydrogenase deficiency. Okay, glucose 6 phosphate dehydrogenase deficiency. Right? Right, right. Yes, 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 very good. Very good, Usman. Yes, biting cells. अच्छा, उसके बाद दो एंजाइम्स मुझे और डिस्कस करने हैं, दो एंजाइम्स मुझे और डिस्कस करने हैं, which I haven't mentioned, but yes, my students are very much, yes. Now beta, from ribose 5 phosphate to pseudoheptulose, okay, this all reactions, okay, from this whole reaction, basically they require an enzyme that is called as trans ketolase, okay, and from heptulose to erythrose 4 phosphate, remember beta, there is an enzyme that is called as aldolase, okay, aldolase, because you already know fructose, yeah, fructose is a keto derivative, and erythrose you already know, yeah. So yes, we have trans ketolase and trans aldolase. Now what is the significance of this trans ketolase? Now trans ketolase, now this trans ketolase beta, now this trans ketolase basically requires thymine. Okay, now you guys are going to tell me thymine is which vitamin? Thymine is which vitamin, guys? I want everyone to answer this question in the chat box. Thymine is which vitamin, and this requires thymine, or thymine as its coenzyme. Okay, coenzyme. Okay, थाइमीन, बाकी सब बताओ. विटामिन बी तो ठीक है आशीष, कौन सा वाला विटामिन है? B1, B2, B3, B4 बताओ. B12 तो नहीं है. सब एक जैसा आंसर करते हैं, बिल्कुल देख लेते हैं कि कोई एक आंसर है. क्या पता उससे टाइपिंग मिस्टेक हुई हो, तो सब लोग मतलब बिल्कुल एकदम धड़ल्ले से आंसर कर देते हैं वही. B1 है बेटा. संजय कृति वेरी गुड. आशीष वेरी गुड. आशीष वाज द फर्स्ट वन, नो, मेडिक्योर वाज द फर्स्ट वन टू आंसर दिस क्वेश्चन. यस. नाउ बेटा, अगर मुझे थाइमीन स्टेटस देखना है बॉडी में, okay, now what is the clinical correlation that is associated with it? Beta, remember we have erythrocyte transketolase, okay. So RBC transketolase, basically it is the sensitive indicator of thymine status. It is the sensitive indicator of thymine status, right? thymine status. Now, apart from that, beta, if there is Wernicke's Korsakoff syndrome, okay, there is a syndrome named as Wernicke's Korsakoff syndrome. Okay, what will happen, beta? What happens in this syndrome? What happens in this syndrome? It is deficiency of thymine. Okay, vitamin B1. We have thymine deficiency. Thymine deficiency. Yes, and the enzyme, yeah, which is deficient over here. You can remember it as transketolase deficiency. So remember about this, beta. This is something which is very, very important to access the indicator of thymine in the body. Okay, that is erythrocyte transketolase. Okay. Apart from that, this transketolase basically requires thymine as its coenzyme. And yes, if we talk about Wernicke's-Korsakoff syndrome, we have thymine deficiency. It is vitamin B1 deficiency, beta. Okay, and the enzyme deficient over here, it is the transketolase deficiency. Transketolase deficiency. Okay. Now, I have a question to everyone over here. I have a question to everyone over here. Okay. Now, you guys are going to tell me the answer. Question number 1. Okay. The oxidative phase of HMP pathway. The oxidative phase of HMP pathway operates in all except RBC, lens, adrenal medulla, and D is adipocytes. Chalo. Let's see how many of you are able to answer this question. Yes. So Ashish says D. No, beta. Ash C. Jainam C. Okay, terrorist says A. No, beta. Chirag Lalwani C. Why C, beta? What is the correct answer then? अगर C नहीं है तो क्या होगा यहाँ पे? अगर मुझे इसी को करेक्ट करना हो, सी ऑप्शन को अगर मुझे करेक्ट करना हो, इफ हैव टू लाइक करेक्ट द सी ऑप्शन, व्हाट इट कुड बी? चलो आरबीसी में होता है मान लिया, लेंस में होता है, ट्रांसपेरेंसी ऑफ लेंस को मेंटेन करता है एनएटीपीएच, ठीक है, एडिपोसाइट्स में भी होता है, ठीक है, यस, इट इज बेटा कॉर्टेक्स नॉट द मेड्यूला, इट इज द कॉर्टेक्स not the medulla, and hence the answer is C. Oxidative phase, I have already mentioned, yes Ashish, very good, it is the adrenal cortex, adrenal medulla नहीं है. देखो हल्का सा चेंज किया और तुम लोग बिल्कुल घूम जाते हो, बिल्कुल पता है क्या होता है कि यू हैव टू डू प्रैक्टिस बेटा. ओके, यू हैव टू डू प्रैक्टिस. ओके, चलो, मैंने कॉर्टेक्स बताया था ना बेटा, अगर आप ध्यान से देखोगे मैंने आपको कॉर्टेक्स लिखवाया है. If you can see beta over here also, like I have not mentioned over here, yeah, you can also write adrenal cortex, right? It is adrenal cortex, right? Adrenal medulla नहीं है बेटा, it is adrenal cortex, right? देखो अब होता क्या है ना कि इसमें थोड़ा सा आपको ध्यान देखना पड़ेगा कि एड्रिनल कॉर्टेक्स में स्टेरॉइड हार्मोन बनता है. तो मैंने जब स्टेरॉइड्स लिख दिया है तो ये अपने आप अंडरस्टूड है कि स्टेरॉइड हार्मोन सिंथेसिस जो है इट हैपेंस इन द एड्रिनल कॉर्टेक्स, right? So that is something that you have to remember, apart from that, one more question that I wanted to discuss. Okay, one more question, you guys are going to answer this. Enzyme in irreversible phase enzyme in irreversible phase of HMP pathway. Remember, we have transketolase, trans-aldolase, epimerase, and 6-phosphogluconate dehydrogenase. चलो, I want the answers from everyone. Yes, Harsh. Now, अब देखो कितनी जल्दी से आंसर है. Harsh, very good. Ash, very good. Jainam, very good. It is the D. ये D आंसर हो गया. चलो, ये D आंसर हो गया. Very good, very good. Everyone is right. Everyone is right. Okay. Usman, Lohar, Medicare, Chirag, Collins. Very good. Very good. Now, again, next question. और इसको थोड़ा सा ध्यान से करना है. The NADPH has all functions except enoyl reductase, HMG coenzyme A reductase. Okay, we have reductive amidation, and the fourth one, it is generation of ATP. चलो बताइए आंसर. बताइए, everyone just try to answer this question. Ash says option D. Okay, symmetry also says option D. What about rest, beta? Medicure, no ATP. D. चलो बेटा, you already know NADPH मैंने लिखवाया था reductive biosynthesis. Okay, reductive biosynthesis of fatty acid, steroids, hormone. ये statement देखो यहीं पे लिखी हुई है. It helps in the reductive biosynthesis of fatty acid, steroids, cholesterol. तो ये सारी जो एंजाइम्स है हम लोग बाद में पढ़ेंगे. एचएमजी कोए रिडक्टेज कोलेस्ट्रॉल वगैरह में, एमाइडेशन रिडक्टेज, देखो सब में रिडक्टेज आ रहा है. तो ये रिडक्शन में तो हेल्प करेगा, ठीक है, बट इट विल नॉट हेल्प इन जनरेशन ऑफ एनी एटीपी. इट विल नॉट हेल्प इन जनरेशन ऑफ एनी एटीपी. That is something you have to keep in mind. That is something you have to keep in mind. So yes, I hope this is clear to each one of you. I hope this is clear to each one of you, beta. Just give me a thumbs up in the chat box. I hope this is clear to each one of you. Yeah. Ashish, very good. Ash, very good. Harsh, Jainam, very good. Symmetry, very good. Very good, beta. Yes. Okay. So, yeah. We have discussed this HMP pathway. Okay. The site, the significance, the phases of HMP pathway, oxidative and non-oxidative. Oxidative phase, we are getting NADPH. I have discussed all the clinical important points regarding NADPH beta. Okay, which organs are responsible for, like, where this reductive biosynthesis is occurring. Okay, the oxidative phase is occurring. What is the function of this NADPH? Okay, and how in the non-oxidative phase also, like, pentoses are generated, and these pentoses are used for the synthesis of RNA and DNA. Okay, and apart from that, the clinical correlation, that is the G6PD deficiency, which are aggravated by some drugs and beans. Hence bodies in RBC, that is again a feature of G6PD deficiency, right? And transketolase, I have already told you about. This requires thymine as its coenzyme. And apart from that, I have discussed all the MCQs, right? So yes, let's end the session for today, and tomorrow or day after tomorrow, I will be just completing with the carbohydrates part. Okay. So yes, please start reading from the book as well. Any textbook you feel like, please start reading from textbook also, and solve as many MCQs as you can. Okay, solve as many MCQs as you can. Right? Sure. Goodbye everyone. Yes.